Hi everyone, we're going to go through the questions that we did today, Friday, February 5th in class. Okay, so the first few questions we did were about whether these different statements that are introduced are kind of talking about a genetic difference between organisms or a non-genetic difference between organisms. And so what I want to point out here is that differences between organisms, whether that's between different people, between different species, between different members of another species, those differences can be due to genetic differences, or they can be due to non-genetic differences. And we agreed that it's always kind of complicated, but that we want to get in the habit of, when we see a phenomenon, either thinking, oh, we're talking about here that the genes make the difference, or we're talking about, oh, the environment, not the genes, make the difference. Okay, so the first one is, many studies suggest that smoking increases incidence of lung cancer. Okay, so now we're talking about differences between people, where some people have lung cancer and others don't. Well, what are we saying drives that difference? What are we focusing in on here? We're saying that difference is in large part attributable to smoking, to exposural, to environmental, to non-genetic differences. So here we're emphasizing, that should say letter B. We're emphasizing a non-genetic difference, right? A non-genetic difference between individuals. Okay, here's a second case. It's the case that people of European descent have higher rates of multiple sclerosis. And that's both the case in America and in Europe. Okay, so now we're talking about differences in terms of chance of developing multiple sclerosis. And we're saying that different populations have different experiences, different rates of multiple sclerosis. Well, this could either be because, say, there's some toxin in Europe that leads to a higher rate of multiple sclerosis, or it could be because of differences in genes. However, we say that people of European descent, even when they live in America, whether they live in America or Europe, they're more likely to develop multiple sclerosis. So this suggests that it's not the environment, because we see this across environments. Instead, it suggests that it's a genetic difference, that Europeans are genetically more likely to get multiple sclerosis. Let's have a look at another one. So there's this gene in which a mutation causes some people to taste a soapy taste when they eat cilantro. Okay, so there's any number of things that are going to influence whether or not you like cilantro, but here we're talking about a single mutation that leads to the specific experience of it where you feel like when you eat cilantro that it's soapy. And we're emphasizing that it's a genetic difference. Okay, so people are different. Some people like cilantro. Some people think it tastes like soap. What's the difference? What we're talking about here is a difference in genes. So here we're focusing in on a genetic difference between people. Okay, so in that case we saw one non-genetic difference and two genetic differences. So let's move on to development of male versus female physical characteristics. So in mammals, as we know, most individuals with physical male characteristics are XY, and it turns out that it's actually the presence of the Y chromosome that leads mammalian embryos to physically develop as males. And so here, what we're emphasizing, no matter how complex the overall case is, what we're emphasizing is a difference in the genes, that some organisms have a Y chromosome and some do not, and that's driving the phenotypic difference that we're talking about. And so here we're talking about a genetic difference. Okay, now let's look at the case in reptiles. So in reptiles, it turns out to be the temperatures that you experience during development leads to whether you develop physical male or physical female characteristics. And so in this case, though there could be other contributors, what we're emphasizing is a non-genetic and environmental cause. That the difference between these organisms in terms of phenotype is largely due to their environment, not largely due to their genes. Okay, so here's kind of continuing along this with reptilian embryos. So now we're also talking about the environment, about environmental temperatures. But now we're talking about how this causes differences in gene transcription and translation, and that influences male versus female development. So here, we're still talking about the environment, but at the same time, we're also talking about genetic characteristics. So here we're talking about both genetic and non-genetic contributors to difference. Okay, so let's switch over to another question, which is the thinking about nucleotide and gene structure. So our first question is, how is RNA transcribed? Is it 5' to 3'? Is it 3' to 5'? Is it both? Or should we not think about it this way? So I'm sure you can guess that I'm thinking of D. So remember, I've said that it's kind of confusing because from one perspective, RNA is transcribed 5' to 3'. From another perspective, it's transcribed 3' to 5'. Depends on whether you're talking about the RNA or the template strand of DNA. And so I've suggested that instead we talk about this stuff in terms of that nucleotide strands add nucleotides on their 3' end, or that nucleotide strands grow on their 3' end, and that this can hopefully help us to clear up these confusions. Okay, so our next question is, what does the RNA polymerase transcription machinery do when it hits a stop codon? I'll give you go ahead and pause it if you'd like to take a second on this one. Okay, so a lot of us would like to say that it stops because it's called a stop codon, but the key here is a codon. So remember, a codon is a triplet that gives that encodes an amino acid that specifies that an amino acid should be added to an amino acid chain, a protein, during translation. So here we're talking about translation, about making proteins. We're not talking about transcription. So what that means is, in the DNA, when the transcription machinery hits a stop codon, say a TAA, to the transcription machinery, that's just 3 nucleotides. It's not looking for stop codons. What is it looking for? It's looking for a terminator sequence to terminate RNA transcription. So when it hits a stop codon, it's just going to continue on through. Rosie Redfield has a nice thing about this, if you want to go back and revisit that. Okay, so our next question is, what does an mRNA start with? So a lot of people are inclined to think that an mRNA starts with an AUG, but let's revisit our structure of the gene. So here we have the transcribed region of a gene. So from left to right, we'd say this is the transcription start site and end site. And then the translated region is here. Now remember, the AUG, that marks the beginning of the translated region. But the whole mRNA includes both the translated region and these regions outside, which are the UTRs, the untranslated regions. So in fact, an mRNA starts with a UTR. It does not start with an AUG. Okay, let's do another one. What do we call a region of DNA that is copied into RNA and which is then translated into protein? I'll give you a sec on that. Go ahead and pause it if you'd like. Okay, so here what are we talking about? We're talking about a region that is transcribed, so somewhere within this red, and then translated, the blue. And so actually we just call that the translated region, right? Because you can't be translated unless you're transcribed, because only mRNAs get translated. So it makes sense to just call this the translated region. Okay, now let's do the other one. What do we call a region of DNA that is copied into RNA but is not part of the sequence that's represented in the protein, not part of the sequence that's translated? I'll give you a second to think about that. Go ahead and pause it. Okay, so here we're talking about the region that is transcribed, so it's part of the red, but it's not part of the blue. So it's within this region but not within this region. And so that here we're talking about these gray boxes, the untranslated regions is what we call them. So we were looking for answer B. Sorry, we kind of did this one before, the translated region. Okay, so now we're going to go through some mutations and what we expect the effect of these mutations to be. So here's our gene structure. We have our regulatory sequences. That would be like the transcription factor binding sites, like what we talked about where OCT1 binds and silences lactase. So there's regions that control whether the gene is on or off. Then we have the promoter, which specifies, basically, it tells us where the beginning of transcription should be. Then we have our transcribed region, our translated region, and our beginning, our start codon and our stop codon, and our UTRs. Okay, so now let's go through some mutations that we can imagine and how we expect that to influence transcription and translation. Okay, so first thing is, what would happen if our ATG were to mutate to an AAG? So our translation start codon is now gone, and it's mutated to an AAG. So do we expect both RNA and protein to be produced from this gene? RNA but not protein, protein but not RNA, neither? Or do we expect them both to be produced but in some kind of strange affected time? Sorry, that went forward despite me. Okay, so I'll give you a second. Go ahead and pause it if you'd like to think about it. All right, so in this case, first we want to ask, is transcription going to proceed? So what directs transcription? Well, the presence of a promoter. We do have a promoter, so we do expect transcription. Then our second question is that mRNA that's produced going to be translated? And so there our question is, do we have a translation start site? So here we do not. So we do expect transcription, we do expect RNA production, but we do not expect translation. We do not expect protein production. So the answer that we're looking for here is that RNA will be produced, but protein will not be produced. That is letter B. Okay, let's think about a different kind of mutation. So what if we have a mutation that disables the promoter? All right, so we work it through again. So now the first thing we ask is, are we going to have transcription? Well, the promoter is the sequence that directs the transcription machinery to initiate transcription. So if we have a disabling mutation in our promoter, if we do not have a functional promoter, then we do not expect mRNA transcription. Okay, so if we don't have transcription, do we expect protein? Well, remember that protein is made by translating RNA, so if we're not making RNA, there's no way we're going to make protein. So under these circumstances, we expect neither RNA nor protein to be produced. All right, let's do another one. So what happens if we have a mutation in the 5' UTR? Okay, so again, our first question is, do we have an intact promoter? We do. So we expect that we will have transcription. And then our next question is, so we have an mRNA. Do we have an intact translation start site? We do, in fact, because the mutation is neither in the promoter nor in the translation start site. And so what do we expect? Well, we expect to have transcription and to have translation. So in fact, we expect that both RNA and protein will be produced. Now, it could be the case that this is a particular mutation within the UTR which affects things. But generally, a mutation in the UTR, we do not expect to affect production of RNA or of protein. Well, what about in the 3' UTR? Well, in this case, the reasoning is exactly the same. We still have a promoter, so we expect mRNA transcription, and we still have a translation start site, so we still expect protein production. All right, let's do another one. Well, what if we have a mutation in the regulatory sequence? Okay, so we have a promoter, so that means we are capable of making mRNA, and we have a translation start site. So when we transcribe RNAs, we are capable of making protein. So what is this mutation going to affect? Well, remember that these regulatory sequences are places where other factors bind and influence not whether we can make RNA and protein, but when. So the example we've gone through the most is the example of OCT1 binding to lactase. And we said if we have a mutation in the regulatory sequence, in the sequence that OCT1 binds to, that this influences when lactase is on, right? So in some people, lactase is on into adulthood because of a mutation in a regulatory sequence. So in general, when we have a mutation in a regulatory sequence, what we expect is that we can make RNA and we can make protein, but that they'll be produced at unexpected times, right? That lactase will be produced at adulthood, that the control of when it's made or the circumstances under which RNA and protein will be made will be changed. So I hate to use wrong here. Obviously, we all have mutations. It's not a question of right and wrong or better and worse. But the point being here that it will change the timing of production of mRNA and protein. Okay, and let's do one more. So what happens if we expect a mutation in the stop codon? Okay, so again, we're going to reason this out. We have an intact promoter, so we do expect transcription. We do have an intact translation start site, so we do expect translation to proceed. It will start here, and it will translate 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3. So what will happen is when it hits this stop codon, well, it's not a stop codon anymore. So instead of translation stopping here, it's just going to continue translating what used to be the UTR, which now is part of the translated region, until it hits the next kind of random, just by chance, the next stop codon down here. So in fact, we do expect that RNA will be produced. We do expect that protein will be produced. But we expect it to be an altered protein. And how will it be altered? So translation will continue. The protein will be longer than we expect. Okay, so our third topic for this video is, or maybe our fourth, is to get back to lactase, and we're going to analyze what happens in this kind of a regulatory relationship when the regulator is mutated. So in class we talked about what happens when this regulatory sequence next to the regulated gene is mutated, but now we're going to talk about what happens when the regulator itself is mutated. Okay, so to remind you, in childhood, the OCT1 gene is off, and the lactase gene is on. And then in adulthood, the OCT1 gene starts expressing mRNAs, making proteins. And those OCT1 genes come down and bind next to the lac gene and turn off the lac gene. So we have lactase in childhood, but not in adulthood. Okay, so let's imagine what happens if we have a mutation in the OCT1 promoter so that OCT1 can never be produced. So what do we expect? Go ahead and pause it if you'd like a minute. Okay, so in this case we have no OCT1 production. So that means in childhood, as is expected, we have no OCT1 expression. But then actually adulthood is going to look similar to this, right? We still won't have OCT1 production, and therefore we will never silence lactase. And so we expect that lactase will be expressed both in childhood and in adulthood. So in this case, we expect that lactase will always be produced. Okay, so in the next case, we want to imagine, so remember that OCT1, we've learned, is on only in adulthood, not in childhood. So that means that OCT1 itself is regulated. We haven't learned about exactly how, but we've learned that OCT1 is regulated in some way. So what happens if we have a mutation in this sequence? So let's imagine two different mutations. So one would be, what if there's a mutation in the regulatory sequence, in this yellow box, such that OCT1 is always produced? Go ahead and pause the video if you'd like to take a minute. So if OCT1 is always produced, well, what does OCT1 do? Well, remember, OCT1 comes down and it shuts off, it binds to lactase and it shuts it off. So if we have a mutation such that OCT1 is always produced, then we expect that lactase will always be silenced. It will be silenced in childhood as well as in adulthood because of this overactive OCT1. Okay, let's now consider a different kind of mutation. So what happens if there's a mutation in those regulatory sequences so that OCT1 is never produced? Go ahead and pause it if you'd like a moment. So actually, we went through a case like this, right? We imagined what would be the case if OCT1 was always off in childhood and in adulthood. And what we reasoned was that if we never have OCT1, then we never turn off lactase. And so lactase is always on. And so under these circumstances, if OCT1 is never produced, then lactase is never silenced, and so now lactase is always produced. Okay, thanks very much. I hope you got something out of that.